Of all the major topics in astrobiology, perhaps the most widely discussed is whether or not Mars is inhabited. To get insight into this age-old question, we can start by looking at the red planet itself. Mars has earned this nickname thanks to the abundance of iron oxides such as hematite on its surface. Apart from iron-based minerals, sulfate-based ones such as gypsum are also quite common. Basically, the surface is highly oxidizing. Mars has a thin atmosphere, with an average surface pressure of 600 pascals, with the highest pressure being 1,155 pascals in the Hellas Planitia, and the lowest being 300 pascals at the summit of Olympus Mons. For some perspective, the Earth's atmospheric pressure at sea level is 101 kilopascals. The atmosphere itself is 95% carbon dioxide, 2.5% nitrogen, 2% argon, and 0.5% trace gases, mostly water vapor, but also oxygen, carbon monoxide, hydrogen, and methane. The average surface temperature of Mars is negative 65 degrees Celsius, with a maximum recorded temperature of 21 degrees near the equator and a minimum of negative 140 degrees Celsius near the poles. Both the north and south poles are covered by a layer of water ice, and there is a layer of carbon dioxide ice which permanently covers the south pole, while this second layer only covers the north pole during its winter. Overall, modern Mars can be described as a frigid, hyper-arid, and hyperlithotrophic world, i.e., very inhospitable to life. However, Mars wasn't always like this. Like the Earth, the geologic history of Mars is divided into stages. The first is the pre-Noachian, which spans from 4.6 to 4.1 billion years ago and roughly corresponds to the Hadean eon of Earth. The next, the Noachian, spans from 4.1 to 3.7 billion years ago and can be seen as analogous to the Eoarchean era. After that, we have the Hesperian, which would be most like the Paleoarchean on Earth. Traditionally, the end of the Hesperian was set at 3.2 billion years ago, but many planetary scientists say it should terminate at 2 billion years ago for reasons we'll discuss later. The final stage, which continues to this day, is the Amazonian. Mars, like the rest of the planets, formed from the protoplanetary disk 4.6 billion years ago. If you look at the topography of Mars, one thing that stands out is that the average elevation of the southern hemisphere is 5 kilometers greater than the northern hemisphere. One hypothesis is that Mars was struck by a giant impactor near the North Pole 4.48 billion years ago, with the northern lowlands being a giant crater, but this is disputed. Between 4.3 and 4.4 billion years ago, the surface of Mars would have cooled sufficiently for liquid water to exist. Despite how arid Mars is today, during the Noachian it was a wet world. We see many weathering patterns from Noachian age sediments in Mars, along with rivers and deltas. Lakes also frequently formed in impact craters, and an ocean known as Arabia may have existed in the northern lowlands, but this is disputed. All of this indicates rain was frequently falling on Mars during the Noachian. Sediments and rocks from this time period cover roughly 45% of the surface of modern Mars, mostly in the southern highlands, including the Noachus Terrae, from which the Noachian gets its name, but also some areas in the northern hemisphere such as the Tempe and Xanthe Terrae. One thing that stands out is that Noachian age surfaces resemble that of the lunar highlands, with many craters, implying that asteroid impacts were the dominant surface forming process at this time. Phyllosilicate minerals such as montmorillonite were the most abundant on the surface, suggesting alkaline conditions were present. The Tharsis Bulge, the large plateau upon which Olympus Mons now rests, formed during this time, and the volatile chemicals it outgassed heavily influenced the composition of the ancient atmosphere. While the modern Martian atmosphere is very oxidizing, the Noachian atmosphere was reducing, with large quantities of methane and ammonia. This would have had huge implications as to how life could have begun on Mars. For one, it means that on Mars, Miller-Urey type reactions would produce biomonomers during thunderstorms. Apart from electrochemistry, photochemistry would likely produce a wide variety of organic molecules in a more reducing atmosphere from methane, carbon dioxide, ammonia, and hydrogen sulfide. High in the atmosphere, UV light would strike these molecules, producing free radicals and resulting in the production of various organic molecules, which would in turn form a condensate haze high in the Noachian atmosphere. This layer would be similar to the one on Saturn's moon Titan, and would block much of the UV light emitted by the sun from reaching the surface, making it more habitable. But some UV radiation from the frequent solar flares of the young sun would reach the surface. Here, various zinc-based minerals could catalyze the synthesis of large organic molecules from gaseous precursors. The reason zinc could facilitate these reactions is because after being struck by a photon, it will have electrons in an excited state for a longer time than other metals. In soils from Gale Crater, which formed during the Noachian, the zinc concentration is 320 parts per million, while concentrations in excess of 130 parts per million are seldom observed on Earth. Furthermore, microporous aluminosilicate minerals known as zeolites could catalyze the synthesis of small organic molecules such as acetate from methane and carbon dioxide. A photon with a wavelength of 2,217 nanometers, which falls in the infrared range, would have enough energy to power this reaction. While a surface probe has yet to identify a sample, spectral analysis from orbiters have suggested zeolites are present in Martian dust. Apart from directly driving organic synthesis reactions, UV light and solar wind particles could prime smaller molecules in the atmosphere. Methane, like all other alkanes, is relatively inert, so if it reacts with a hydroxyl radical produced by interactions between UV light, water, and transition metals, it will result in the production of methanol, formaldehyde, and formic acid. Since these compounds are more reactive, they can be fed into prebiotic synthesis pathways. This brings me to a key question. How could life have begun on Mars? One might think that with Miller-Urey type reactions taking place, you would have had a primordial soup on ancient Mars. However, even if this was the case, it couldn't give rise to life on its own because a primordial soup is in equilibrium, while life requires a disequilibrium to drive its metabolism. There is also the tar problem, one in which Miller-Urey reactions produce a largely random assortment of medium-sized organic molecules, many of which will competitively inhibit biomonomers from reacting with each other and giving rise to life. As such, a hydrothermal system would be a more likely candidate for the cradle of life on Mars. The high temperatures and minerals present in them will readily facilitate pre-metabolic reactions. While abiotic processes will produce biomonomers on ancient Mars, most of the organic matter will be in the form of small 1 to 3 carbon molecules. When these go through various synthesis pathways, a much greater portion of the organic carbon will be in the form of biomonomers, and most of these will be the ones the first organisms would use, negating the tar problem. Furthermore, hot, cold, wet, dry, and acidic basic cycles are key drivers of prebiotic chemistry, and these are staples of terrestrial hydrothermal systems. But if life on Mars did come about, would a volcanic or serpentinizing hydrothermal system be a better candidate for its place of origin? The reason most astrobiologists think serpentinizing systems are a better candidate for the cradle of life here on Earth is because a wide variety of organic molecules are produced via Fischer-Tropsch type synthesis reactions in them, while only methane has been produced in volcanic vents. However, the more reducing atmosphere of ancient Mars would mean there were more organic molecules in the surrounding environment, negating the need for organic molecules to be produced from inorganic carbon in the hydrothermal system itself. Furthermore, serpentinizing systems are fairly stable, while volcanic hydrothermal ones have far more variation in activity levels, leading to much more rapid fluctuations in pH, temperature, and hydration. So, volcanic hydrothermal systems would arguably be a better candidate for being the cradle of life on Mars. But if life did begin on Mars, in what ways might it be similar or different to life on Earth? Well, for one, during the Eoarchean and Noachian, meteors would have fallen to the surfaces of Earth and Mars regularly. The metallic ones would have contained schreibersite, an iron-nickel phosphide mineral. When schreibersite is immersed in water, it will corrode and release reactive phosphorus species, which can then phosphorylate organic molecules in a manner similar to how ATP does this in our cells. So, Martian biochemistry may also be powered by organophosphate groups, with its own version of ATP, which acts as life's energy currency. Given that we've identified abiotic analogs of the TCA cycle, glycolysis, and pentose phosphate pathway, it's possible that a hypothetical Martian organism would employ these pathways as well. Since the D isomer of sugars and the L isomer of amino acids, the stereoisomers we use, are more abundant in the solar system, it would also be likely that a Martian organism would use the same chirality as life on Earth. Lastly, since Mount Morillonite clay is present in Noachian age deposits, Martian life would also likely use RNA. But how could life be different on Mars? Well, for one, with an abundance of organic carbon in the environment, Martian life wouldn't have needed a carbon reduction pathway from the outset like the Wood-Young-Dahl pathway. Many enzymes in the Wood-Young-Dahl pathway have iron-sulfur clusters, which closely resemble minerals which facilitate Fischer-Tropsch type synthesis reactions in hydrothermal systems. Given the importance of photochemistry in a reducing environment, the deep-rooted enzymes of Martian organisms could instead have zinc or aluminosilicate/zeolite cofactors. Finally, with a greater assortment of amino acids, sugars, and nucleotides being produced by Miller-Urey type reactions in the Noachian atmosphere, Martian life could end up incorporating a wider variety of these monomers in its biochemistry than life on Earth. But how would these differences in fundamental biochemistry impact the hypothetical ancient Martian biosphere? If we look at the contemporary Eoarchean of Earth, the only biophilic element with an isotopic signature of being metabolized is carbon. Similarly, on the Noachian Mars, life could start by uptaking small organic molecules and feeding them into synthesis pathways driven by largely abiotic mechanisms to produce biomolecules. This synthesis could be driven by the hydrolysis of Schreibersite-derived reactive phosphorus species, secreting orthophosphate as a waste product. As life began to develop its own biocatalysts, be they enzymes or ribozymes, it could then incorporate the aforementioned zinc and zeolite cofactors, which would allow it to produce organic carbon in the form of acetate from methane and carbon dioxide to be assimilated. This would be powered by light, and since it's easier to photochemically combine carbon dioxide and methane than it is to produce carbon dioxide using water, iron, or sulfur as the electron donor, phototrophy could very well have evolved on Mars before Earth. While the first organisms could use Schreibersite-derived reactive phosphorus species like pyrophosphate to synthesize biomonomers from simple organic precursors, such a pathway could be reversed, breaking down abiotically produced biomonomers into simple organic molecules to produce reactive phosphorus species to power the organism's metabolism. Instead of producing a pyrophosphate, it could add a phosphate group to an organophosphate via a phosphoanhydride bond. This molecule would act as the energy currency for Martian life, kind of like how life on Earth uses ATP. Initially, such a pathway would metabolize the biomonomers used by the organism, which were produced by Miller-Urey type reactions. Later, it could evolve the ability to break down similar molecules that aren't used in the organism's biochemistry, and in so doing, taking advantage of the tar problem. So, the more reducing environment would be more conducive to the genesis of fermentation than on the early Earth. As the early photomethanotrophic organisms evolved the ability to produce their cofactors from simpler and simpler starting materials, and the fermenters evolved the ability to break down biomolecules into simpler and simpler organic molecules, they could both begin to venture out of their hydrothermal cradle. Given that during the Noachian/Eoarchean, meteors fell regularly, a question that comes up is, could life from one planet have been blown off into space and land on another? Well, experiments have consistently shown that microorganisms could survive the shock associated with a meteorite impact. Furthermore, there is some evidence that the outer membrane and peptidoglycan layer in bacteria is the remnant of a dormant spore, which the last universal common ancestor of all life possessed. However, the evidence is far from conclusive, and being able to form a dormant spore doesn't necessarily mean it could survive in the irradiated vacuum of space, although modern endospore-forming bacteria can. But if life did get transplanted from Earth to Mars and survive, what kind of challenges would it face? Well, for one, if an organism from the Eoarchean Earth landed on Noachian Mars, it would have a large quantity of organic carbon available to it. However, there will be abiotically produced compounds with similar chemical properties to the molecules it uses, which can competitively interact with them, i.e., it's going to run into the tar problem. Conversely, with an abundance of organic molecules on Noachian Mars, organisms there would be less likely to have evolved a mechanism that can reduce carbon dioxide, and thus wouldn't be able to survive on the relatively organic poor early Earth. Another problem that would go both ways could be auxotrophy. A protrophic organism can synthesize all of its essential nutrients, while an auxotroph has to get them from the environment after they were synthesized by another organism. These nutrients are often enzyme cofactors, also known as vitamins or amino acids. Early on, most organisms likely depended on abiotic processes in the environment to produce these before they evolved biosynthesis pathways to make the nutrients themselves. These external reactions would have been environment specific, so if the organism was suddenly transplanted to a different environment, it couldn't survive. But if panspermia from Earth to Mars or vice versa occurred, how could we tell? Well, if we found Martian life, without a comprehensive understanding of what ways biochemistry changed from the first organism to the last common ancestor, there is no way to rule out panspermia completely, so long as the Martian life is carbon and water based. However, if we find homologous genes for enzymes involved in ancient metabolic pathways like glycolysis or the TCA cycle, along with 16S rDNA in Martian organisms, then it's a virtual certainty that panspermia occurred. If they use the same 20 amino acids and 4 nucleobases, that would be good evidence for panspermia, but it wouldn't be conclusive because there are some reasons to believe life anywhere would utilize these, but why that is a story for another day. Part of the reason we think early Mars had a reducing atmosphere is because there would have to be high levels of methane to induce a sufficient greenhouse effect in order for water to stay liquid on the surface. Carbon dioxide wouldn't cut it that far out and with a faint young sun. However, the Noachian is hypothesized to have ended with an ice age, with glaciers covering the southern highlands. This is because the erosion patterns from this time more closely resemble those of glacier formation and melting versus rain, but again, this topic is subjected to an ongoing debate. At the start of the Hesperian, most of the water bodies which were present during the Noachian disappeared, and if the Ocean of Arabia had existed, it was gone by this time. Exactly what happened to the water is a mystery, but in my opinion, it went into hydrated minerals, which incorporate water molecules into their crystal structure, including illite and gypsum. The Hesperian is marked by a shift in surface chemistry, from being dominated by alkaline phyllosilicate-based minerals to acidic sulfur-based ones. There was also a switch in that volcanism replaced impact cratering as the dominant surface-forming process. It may sound like Mars began its time as a desolate, arid world at the start of the Hesperian, but this wasn't to be. Massive eruptions occurred on the Tharsis Bulge, which saw the formation of Olympus Mons and the other major Martian volcanoes. These released huge quantities of water vapor into the atmosphere, which poured down on the eastern side of the Tharsis Bulge in a torrential rain. The water then flowed down into the northern lowlands to form the Deuteronilus Ocean. When you look at a topographic map of Mars, you can see the outflow channels from this in Cassi Vallis and Vallis Marineris. Afterward, Mars experienced sporadic precipitation, but the rate was 1/1000th that of the earlier Noachian. While this was happening on Mars, the last common ancestor of everything alive today lived on Earth, also known as Luca. While simpler than most modern microbes, Luca was still within the range of variation seen in modern prokaryotes. Shortly after, around 3.5 billion years ago, microbes began to form macroscopic structures such as stromatolites along the ancient seashore. So, if life did begin on Mars, it could have reached a level of complexity on par with modern bacteria by the Hesperian, and it could live in the ocean. Now, you may have heard that the Deuteronilus Ocean was too salty for anything to live in. It is true that it was more saline than Earth's oceans, but it was less saline than the Dead Sea, which, contrary to the name, hosts microorganisms. So, even though the Deuteronilus Ocean may have been too salty for life to begin in, it could support life once it arose. In fact, I would argue that the Earth's oceans were too salty for life to begin there. At the start of the Hesperian, phototrophic organisms could survive and grow by combining atmospheric methane and carbon dioxide into organic molecules. Also, with less reactive phosphorus species available from the lack of meteoric Schreibersite, it could use light energy to replenish its cellular energy currency. At the bottom of the ocean, away from the light, Fermenters could break the dead organic matter produced by the phototrophs all the way back down to methane and carbon dioxide, getting all the energy possible out of it. It was around this time on Earth that we find the oldest evidence of methanogenesis. So, we could have a unique version of a carbon cycle, but one that involves carbon in two oxidation states in the gaseous phase versus in the oxidized one along with an electron donor. Later in the Hesperian, the Martian dynamo began to slow down, causing the magnetic field to weaken. This resulted in the solar wind stripping away the atmosphere and would have removed any UV shielding organic haze. As a result, the photic zone would have become uninhabitable, restricting any phototrophs to the shallow seafloor. However, there would be another energy source available to life. The solar wind would not only thin the atmosphere, it would make it more oxidizing, as it selectively blew away the lighter, reduced compounds. The result would be a redox disequilibrium between the mantle and atmosphere life could harvest. Sulfur dioxide released by volcanoes would react with water vapor to form sulfurous acid, which would then undergo photolysis to become sulfuric acid. This would rain down into the ocean, and it, along with sulfate, its conjugate base, could be used as an electron acceptor to oxidize reduced compounds released by volcanic vents, such as hydrogen and methane. It was around 3.5 billion years ago that we find the first evidence of distillatory sulfur metabolism here on Earth. In the latter part of the Hesperian, the surface would start to become more oxidizing, with dust made of iron oxides and perchlorates. When these blew into the nearshore waters, it would make the benthos less habitable, further attenuating Martian phototrophy as a whole. But chemotrophs inhabiting nearshore hydrothermal vents could use the iron oxides to oxidize not only methane and hydrogen, but hydrogen sulfide, and the perchlorates to oxidize all of these along with ferrous iron. As I mentioned before, there is some disagreement as to when the Hesperian-Amazonian boundary should lie, with some saying 3.2 billion years ago, while others saying it should be at 2 billion years ago. This is because the Deuteronilus Ocean, along with all other major standing bodies of water on Mars, had dried up by 3.2 billion years ago. However, over the span of the next 1.2 billion years, we still see evidence of fluvial activity on the red planet, largely driven by glacier formation, melting, and evaporation. At this time, much of Mars would have resembled places like the Atacama Desert and Antarctica. So, while Mars would have been quite inhospitable around this time, the hardiest microbes on Earth could survive on it, even if Mars never hosted life. This gray area between the wet world of ancient Mars and the unworldly, desolate planet we see today has fueled disagreement as to when the quote-unquote modern period of the geologic history of Mars began. Many astrobiologists look at how organisms persist in Mars analog environments to see how habitable Mars is. But, while they can replicate one or two of the hardships there, they can't replicate all of them. And as we discussed in the video about extremophiles, combinations of hardships are what render certain environments uninhabitable. However, many of these analog environments would be a good model for Mars during the early Amazonian. Apart from the Atacama and Antarctica, environments like the Rio Tinto River in Spain can show how microbes survived in ancient Martian rivers. The Uyuni salt flats in Bolivia can act as an analog for the salt flats which stood where the Deuteronilus Ocean once was, and places like the Mepinong Cave in South Africa can provide insight into how ancient Martian life could have begun to colonize the subsurface. In the most desolate environments on Earth, many microorganisms use the atmosphere itself as an energy source. They do this by oxidizing trace amounts of methane, hydrogen, and carbon monoxide, which exist as minor constituents of the atmosphere, while using oxygen as the electron acceptor. During the early Amazonian of Mars, solar wind particles would split water molecules high in the atmosphere. The lighter hydrogen atoms would fly off into space, while the oxygen atoms would recombine into diatomic oxygen molecules. This oxygen wouldn't accumulate in the atmosphere because it would react with various minerals in the crust, but it would still be more prevalent than the aforementioned reduced trace gases. As such, Martian microorganisms from the early Amazonian could use trace gas oxidation to power their energy metabolism along with grow and divide. So far, we've assumed photosynthesis as we understand it, where another element is photooxidized to reduce carbon dioxide to an organic molecule, didn't arise on ancient Mars because of the abundance of methane as a reduced carbon source in the atmosphere. But, with the more oxidizing conditions of the early Amazonian, would water splitting photosynthesis be advantageous to life? Well, over most of the surface of Mars, I would argue it wouldn't, because if any organism was exposed to sunlight, it would also be exposed to intense UV radiation. However, UV light is quickly absorbed by water, so if an organism lived at the bottom of an early Amazonian river, it could get enough light energy to carry out photosynthesis, while being protected from harmful radiation. Now, we come to the Mars of today, the hyper-arid world. Whether or not there are hydrologic processes involving liquid water on the red planet is still an open question. Gully-like formations on the walls of craters may have been formed by flowing water, but other explanations haven't been ruled out. The biggest hindrance for water existing as a liquid on Mars is the low atmospheric pressure. The triple point of water is at 612 pascals. Below this pressure, water cannot be liquid. It will go directly from gas to solid and vice versa. With an average surface pressure of 600 pascals, much of Mars, especially the southern highlands, can't host liquid water. Much of the northern lowlands have a surface pressure above the triple point of water, but even then, it would only be liquid for a few degrees above zero before it boils. Frost has been observed forming on the northern lowlands, so if it melts, it will briefly wet the soil. Perhaps one of the best places for life on Mars, based on this criteria, is Valles Marineris. Its low altitude and equatorial latitude would make both the pressures and temperatures more conducive to liquid water. Furthermore, Valles Marineris is quite humid, and we frequently see it filled with fog, although this fog is water ice versus liquid droplets. Another hindrance for life on Mars is the perchlorates in the soil. Martian soil is usually between 1 and 0.5% perchlorates by weight. For some perspective, the soil in the Atacama Desert is on average 0.02% perchlorates by weight, with the highest concentration being 0.6% in the nitrate ores. Exactly how Martian perchlorates were formed is not comprehensively understood, but the general consensus is that it is photochemical in nature, involving high-energy UV photons, chloride ions, and water. Perchlorates are a highly stable yet very corrosive oxidant, which quickly oxidizes organic matter, making them very toxic to life. Although they aren't all bad, perchlorates act as an antifreeze, decreasing the melting point of water, meaning it could remain liquid for longer. However, there are some bacteria which use perchlorates as an electron acceptor for their respiratory processes. The perchlorate is first reduced to chlorate and then to chlorite. Next, the chlorite is disproportionated into chloride ions and oxygen. But it should be noted that culture experiments with perchlorate respiring bacteria have used concentrations of less than 10 millimoles per liter, while the concentration in any Martian mud would be in excess of 50 millimoles per liter. Could microbes grow in water with perchlorate levels seen on Mars? We don't know, but even if an experiment showed that they could, this still wouldn't prove that perchlorates wouldn't prevent life from existing on Mars, because, as we're finding out, it's combinations of hardships versus a single variable that make some environments uninhabitable. A metagenomic study of the McMurdo Dry Valleys in Antarctica has indicated that they were sterile, with no organisms in them. Like Martian soils, these have high levels of perchlorates. While it may be possible for an organism to survive in a habitat with the same levels of perchlorates, repairing the damage they cause is an energetically expensive process. Given how oligotrophic the dry valleys are, it may be that there just isn't enough energy available to the microbes to repair the oxidative damage faster than they incur it, i.e., the combination of toxicity and oligotrophy makes the dry valleys uninhabitable. Similarly, the high concentrations of perchlorates, along with the scarcity of available energy in Martian soil, can render them sterile. Given the short duration of any liquid water on the Martian surface, if any organism is to live there, it would have to be capable of going dormant to survive the dry periods. Quite a few bacteria here on Earth can form an endospore, a hardy dormant structure to endure periods of hardships. Given how oligotrophic Mars is, any microorganism would have to go through several cycles of dormancy and revival before it could replicate, because it can only assimilate a small quantity of biomass before it has to go dormant again. All this to say that the division time of a Martian microbe would be at the very least centuries, and more often millennia. Once every 3 years, Mars is covered with global dust storms, which would then disperse any organism across the planet. The odds that it would land in a suitable habitat at any given time are quite low, but after enough dispersal events, the odds add up to be much higher. However, dormant spores do have a shelf life due to the radiation present. There are a number of estimates as to how long a spore would last on the Martian surface, and the highest estimates are at 600,000 years. This comes to 200,000 dispersal events, so after all of those, you'd argue that most of the organisms would land at least on one suitable habitat. The problem is that the organism would incur damage to its molecular machinery during dormancy because of the radiation, and given how little energy there is available, it may not be able to repair all the damages before it has to go dormant again, putting the cell into a death spiral. If there is a less than 50-50 chance that an organism will be able to divide before dying, the entire population will slowly decline to extinction. However, long-term geologic processes could send Martian life a lifeline. Mars undergoes ice ages once every 15 to 40 million years, and experiences volcanic eruptions once every 2 to 8 million years. On top of that, meteors fall regularly, and these expose or bring fresh rocks to the surface, which would include sulfide and ferrous iron-based minerals. These could be used as an electron donor to drive the microbe's energy metabolism, and the electron acceptor could either be perchlorates or the trace amounts of oxygen in the atmosphere. The concentration of diatomic oxygen in the Martian atmosphere is 1300 parts per million, with a partial pressure of 1.5 pascals in the regions with the highest atmospheric pressure. The oxygen in the Martian atmosphere is believed to be a byproduct of the perchlorate cycle. When a volcanic eruption brings fresh rock to the surface, or a glacier uncovers them, it could provide life with a hotspot for growth and division, boosting the total population of Martian microbes. Afterward, the population will decline as the organisms have a less than 50-50 chance of surviving to division until another ice age or volcanic eruption. If this is the case, a Martian surface biosphere would be defined by a bloom and bust cycle on geologic timescales. Furthermore, a retreating glacier would not only expose fresh rock, but if it's in a low altitude mid latitude region, it would periodically melt, briefly wetting the sediment at the base. Mars has glaciers today, and in the temperate regions, where temperatures occasionally exceed 0 degrees, it would provide any microbes with a fleeting yet recurring source of liquid meltwater. This brings me to a possible haven for life on the red planet, the cryosphere on the poles. At first, this may seem a bit counterintuitive, because life needs liquid water, and the ambient temperature never exceeds 0 degrees. However, localized heating effects can result in above freezing temperatures. Sometimes, bits of dust are blown onto the glaciers, which are known as cryoconite. When sunlight strikes the dark colored particles, it heats them up, causing them to melt a hole in the ice. The surface will then freeze over, while the light penetrates the ice and heats up the particles, keeping the surrounding water liquid. These cryoconite holes often host a self-contained microbial community. This microcommunity is powered by light, and the members cycle nutrients between each other. Such holes could form on the North Pole Martian ice cap when the water ice layer is exposed during the Northern Hemisphere summer. Furthermore, the bottom layer of the ice sheets could also host life. Ground-penetrating radar from an orbiter has revealed what appears to be a subsurface lake beneath the South Polar ice cap. In order to stay liquid, the water would either have to be hypersaline, being 40% calcium perchlorate by weight, or be heated hydrothermally. If the latter is the case, it could very well be a habitable spot. In Antarctica, there are also subglacial lakes hundreds of meters below the surface of the ice sheet. In these, we found microbial life with physiological features that are telltale signs of adaptations to oligotrophic environments. Most of these lakes are in permanent darkness. However, the layer of ice over one of these lakes, Lake Untersee, is thin enough that large amounts of sunlight pass through. As a result, stromatolites formed by mats of photosynthetic cyanobacteria cover the lake bed. If a portion of the Martian North Pole ice cap was near the edge and heated hydrothermally, similar communities could form. But before you get too excited, I should mention that a reanalysis of the data from the orbiter indicated that the subglacial lakes may instead be a layer of frozen clay. With the prevalence of harmful radiation and toxic chemicals on the Martian surface, perhaps the best place to find life is underground. One of these habitats are long caverns known as lava tubes. These are formed when a magma flow melts its way through rocks and then solidifies. Here, microbes would be protected from the harsh solar radiation and perchlorates, but what energy source could they utilize? One might be trace gases in the Martian atmosphere. As mentioned before, on Earth, there are microorganisms which power their metabolism by the oxidation of methane, hydrogen, and carbon monoxide, with ambient concentrations in the atmosphere of 1.7, zero point 0.5, and zero point 0.1 parts per million respectively. There are 44.8 millimoles per liter of air at sea level, so this comes to 76.2, 21.2, and 4.23 nanomoles of methane, hydrogen, and carbon monoxide per liter. With the combustion of methane, hydrogen, and carbon monoxide yielding 890, 286, and 283 kilojoules per mole, the oxidation of all the trace gases in 1 liter of Earth's atmosphere at sea level would yield 75 millijoules. In the Martian atmosphere, the most abundant of these trace gases is carbon monoxide at 700 parts per million. This is a little more than half the concentration of oxygen, and since one molecule of oxygen will oxidize 2 molecules of carbon monoxide, the reduced trace gases, i.e. the electron donors, would again be the limiting metabolite on Mars as they are on Earth. With the greatest atmospheric pressure on the surface of Mars being 1,155 pascals, there will be at most 511 micromoles per liter. This means there will be 357 nanomoles of carbon monoxide per liter, which would yield 101 millijoules when oxidized. So, trace gas oxidation on the lowest parts of Mars will yield more energy than it would on Earth. In places where the atmospheric pressure is 858 pascals, the energy per liter of air would be the same as it is on Earth at sea level, and where it's 612 pascals, the triple point of water, 1 liter of air will yield 54 millijoules. While microbes could get energy out of the atmosphere, could they also obtain material to produce biomass out of? Well, there's an ample source of carbon in the form of atmospheric carbon dioxide, but biomass is composed of other elements. There is hydrogen in the atmosphere as water vapor and molecular hydrogen at a concentration of 15 parts per million. Water vapor is more abundant, but water is hard to split, so instead, Martian microbes could react some of the carbon monoxide with hydrogen to produce formaldehyde. Since it has the same elemental stoichiometry of organic matter, CN, H2N, ON, the formaldehyde could be fed into biosynthesis pathways powered by the oxidation of other carbon monoxide molecules. Generally, microbes which inhabit the most oligotrophic environments here on Earth assimilate 8% of the organic carbon they metabolize into their biomass versus oxidizing it for energy. And with a ratio of carbon monoxide to hydrogen in the Martian atmosphere being 46:1, the availability of material to build biomass out of versus the energy available would be the limiting factor for any Martian trace gas oxidizers. They would also need to obtain nitrogen, and if microbes are on Mars, they could also get this from the atmosphere in the form of diatomic nitrogen via an analog of diazotrophy. Further down, rocks will be protected from the localized effects of solar heating, and the temperature will constantly be below freezing. However, once you get down to 5 kilometers deep, the temperature rises above 0 degrees Celsius, and the porosity is similar to the Earth's crust at 1.8 kilometers deep. Microorganisms surviving at this depth are well documented, so Martian life could very well have found a refuge deep underground. Subsurface microbes on Earth oxidize reduced metals and minerals as an energy source, and organisms deep in the Martian crust could do the same. These could also use the trace quantities of oxygen in the Martian atmosphere as an electron acceptor. While not as abundant as it is in Earth's atmosphere, the main factor limiting how fast rock metabolizing microorganisms can grow is accessing the elements when they are bound in a crystalline structure. This limitation limits how quickly the deep microbes can divide to once every thousand or so years. So, the lower concentrations of oxygen probably wouldn't hinder how fast rock metabolizing organisms on Mars could grow. However, if we go slightly deeper, things might change. Oscillations in the magnetic field of Mars have hinted at a planet-wide subsurface brine reservoir. While the surface seems almost entirely geologically dead, the Martian mantle is still convecting, and this could bring reduced gases such as hydrogen, methane, and hydrogen sulfide into the brine reservoir. At the very top layer, oxygen could be used as an electron acceptor to oxidize these chemicals, but it would become quickly depleted. Sulfate and ferric iron produced by the oxidation of sulfide and ferrous iron could be used to oxidize hydrogen and methane, but these would quickly become depleted as well. So, further down, something else would have to be utilized. One acceptor that's quite abundant, making up 95% of the Martian atmosphere, is carbon dioxide, which can be used to oxidize hydrogen. However, it can't be used to oxidize sulfides. This reaction produces methane, so it may be that much of the Martian deep biosphere, if it exists, is powered via methanogenesis. Down here, variations in the upper mantle temperature can drive the movement of water in the brine reservoir, and along with it, metabolites, nutrients, and the microorganisms themselves. In the Martian atmosphere, methane levels peak in the late summer and early autumn of the northern hemisphere, while they reach their minimum in the late winter and early spring. It may very well be that this is produced by microbial activity deep underground, but again, other explanations haven't been ruled out. While there are many possibilities for life on the red planet, how could we go about detecting it? The hunt for life on Mars can be said to date back to the observation by William Herschel that the Martian ice caps grew and shrank and the planet had an atmosphere. Based on this, he speculated that Mars was inhabited. In 1877, the astronomer Giovanni Schiaparelli thought he saw channels on the Martian surface. The Italian word for channel is canali, which is mistranslated into canals in English, the implication being that they were artificial structures. This news led to a flurry of speculation as to what kind of advanced civilization exists on Mars. But, apart from having a mistranslation involved, the channels Schiaparelli observed were instead scratches on his telescope lens. Nonetheless, for the subsequent 88 years, many astronomers entertained the notion of a thriving biosphere on Mars. These hopes were dashed in 1965, when Mariner 4 became the first probe to conduct a flyby mission and revealed a cold, desolate world. Yet many held on to the possibility that some kind of hardy life still survived there. In 1976, Viking 1 and 2 became the first probes to successfully land on Mars. To see if the soils were inhabited, they released nutrients with radioactive carbon-14 in them. The reasoning behind this was that if there were microorganisms in the soil, they would metabolize it and in so doing, release carbon dioxide with a radioactive carbon-14, which the probes would detect. When both probes released the nutrients, they got a positive result, but they repeated it a week later and the results were negative. This indicated that the organic carbon was likely oxidized abiotically by perchlorates in the soil. In 1996, there was another flurry of speculation about the possibility of life on Mars. In 1984, the Allen Hills meteorite was found in Antarctica. This rock had originally come from Mars and was dated to 4 billion years ago. When it was put under a microscope in 1996, scientists found structures which looked like bacteria in it. However, these structures were smaller than any bacteria known to exist on Earth, and other explanations couldn't be ruled out. In 2022, pretty much all hopes that these microstructures are biogenic were dashed when it was shown that brine flowing over the rock and into the pores would produce such shapes. Today, Many probes are continuing the search for traces of Martian life. Mars Odyssey, an orbiter launched in 2001, is using radar to search for subsurface liquid water. On the surface, two rovers, Curiosity and Perseverance, have stolen the spotlight. Curiosity was launched in 2011 and landed in Gale Crater in 2012. Gale Crater was formed during the Noachian and, at the time, had been filled with water. The probe is equipped with laser-induced breakdown spectroscopy, or LIBS, which shines a laser on an object to energize the electrons in the surface material and uses the spectrum of the resulting emissions to determine its composition. Using LIBS, Curiosity has identified the presence of chlorinated short-chain alkanes and chlorobenzene in ancient mudstones. While these could have been produced by ancient Martian life, there are many other means by which such organic molecules could be made. Indeed, more complex organic molecules are common in asteroids and comets. LIBS can also analyze the isotopic composition of surface materials to look for signs of life. The other rover, Perseverance, was launched in 2020 and arrived in Jezero Crater in 2021. Jezero Crater is younger than Gale, having formed 3.1 billion years ago, after the oceans had dried up, yet the surface of Mars was still conducive to the presence of liquid water. Perseverance has identified lakebed sediments within the crater itself. Perseverance also has an onboard spectrometer, which can detect organic molecules, and an X-ray to look for petrologic signatures of life. There are also some unmanned Mars missions planned for the near future. One of these is the Mars Sample Return mission, which will land a rocket on the surface to bring samples collected by Perseverance back to Earth to look for traces of life. A similar mission planned by China, which is known as Tianwan-3, will return samples from a yet-to-be-determined location. Japan is currently developing the Milos rover, which will also land at a yet-to-be-determined location. Once there, it will use fluorescence microscopy to look for life and use isochron dating to determine the age of sediments. Lastly, India is working on the Mars Orbiter 2 to investigate the evolution of the Martian crust. While no mission has yet been planned, planetary scientists are currently drawing up concept designs for rovers to explore the polar ice caps and the lava tubes. Now, how can we go about finding traces of life, past or present, in future missions? Well, one thing we can do to start is to look for fossilized macroscopic structures such as stromatolites. The shores of ancient lakes and oceans would be prime spots to look. While such structures may very well be buried, rovers equipped with ground-penetrating radar can search for them. Furthermore, since many photosynthetic organisms form biofilms on streambeds, such a probe could also look for the fossilized remnants of these in early Amazonian streambeds. Although, I think I should add the caveat that determining the biogenicity of structures from the early Earth is quite difficult, even when we have the object in question in a lab. So, even if a probe was to identify stromatolite-like structures buried underground, it would be far from conclusive evidence of ancient life on Mars. We can also look for chemical evidence of past life. As mentioned before, Curiosity shined a laser on ancient mudstones and found chlorinated organic compounds. These may have been produced by ancient microbes, but even if the carbon atoms in them were once incorporated into larger biomolecules, being on the surface of the rock would mean they have been broken down by the radiation perchlorates to the point that they are indistinguishable from organic molecules produced abiotically. However, when the mudstones were mud, microbes or biomolecules produced by them would penetrate into the deeper layers. Once the mud had become lithified, these molecules would have been more protected from the radiation and oxidants on the surface. Another probe could use a drill to extract the samples from the interior of the mudstones and then run them through a mass spec. The molecules astrobiologists would be looking for are lipids, as these last longer than any other biomolecules. If we find varieties of lipids which cannot be produced abiotically, it would be pretty good evidence for past life, and if there were some used by organisms on Earth, it would indicate that panspermia occurred. However, it wouldn't be conclusive because there might be an as of yet undescribed mechanism by which the lipids in question can be produced abiotically. Theoretically, We could send probes to look for isotopic or petrologic signatures of ancient life in the rocks. However, again, determining the biogenicity of isotopic and petrologic signatures from the early Archean is quite dodgy, and that's in spite of the fact that we know how early organisms fractionate various stable isotopes and what kind of petrologic signatures they leave behind. Without knowing how Martian organisms, assuming that they existed in the first place, did this, it would be virtually impossible to use such signatures as solid evidence of life. When it comes to modern Mars, there are several methods we could use to determine if life exists. I mentioned that missions to the polar ice caps and lava tubes are planned. If a mission was to go to the North Pole, it could search for cryoconite holes, and if it finds them, the probe could then drill into one and run the fluid inside through a mass spec. If there are organisms in the hole, it should be quite easy to identify them, because even if they used a completely different biochemistry, they would almost certainly employ some kind of large complex biomolecules that the instrument would pick up. A polar probe could also carry ground penetrating radar or sonar to identify subglacial lakes, and unlike the orbiter, it could tell if they were liquid water or frozen clay. While this initial rover couldn't sample from lakes hundreds of meters under the ice, it could give a reason to send a follow up mission if it identified one. Another surface probe would land on the ice over where the subglacial lake is, and it would release a submarine with a heated tip to melt its way through the ice. The tip would be powered by solar panels on the surface probe, with the electricity being delivered to it via a cable. Once in the lake, the probe could also use mass spec to look for organisms. A probe exploring a lava tube could also use this to find any organism in the walls or the floor of the tube. If a mass spec identifies not only large biomolecules, but specifically DNA with adenine, thymine, guanine, and cytosine, then studying Martian life would get a lot more straightforward because we could use whole metagenome shotgun sequencing in follow-up missions. This is where you sequence all the DNA of all the organisms in the specific habitat. While we wouldn't have a reference like we do here on Earth, where we can say that X enzyme carries out Y process, it would allow us to determine how different microbial communities in various Martian environments are. I.e., we could determine if the microbial communities in lava tubes, on the surface, and in cryoconite holes are distinct from one another, or if dust storms homogenize the biosphere via principal coordinate plots. While we could tell if different biomolecules are used in different habitats, we could not tell if this is because different operational taxonomic units are present in them, or if the same ones are expressing different genes in different environments. This brings me to another important aspect of finding life on Mars, making sure that the probes we sent there aren't carrying earthly organisms. Currently, there are international treaties which ensure that any probes sent to Mars are completely sterile, and this is accomplished by heat treating them at 114 degrees. If we find organisms on Mars, we would likely try to bring some back here to Earth to study them in a lab, and hopefully get them to grow in pure cultures. This would allow us to study their cellular and molecular biology along with their physiology in more detail. Some might worry that Martian microbes could be pathogenic and cause a sci-fi style global pandemic. I would say this is extremely unlikely for two main reasons. First, any Martian microbes would be so case selected due to the oligotrophy of their environment, they would divide far too slowly to infect someone. Second, it's virtually impossible for pathogens, which infect distantly related organisms on Earth, i.e., plants, to infect humans because of the differences in biochemistry. So, a Martian organism would have an even harder time developing the necessary adaptations to infect human tissues. But as mentioned before, the surface of Mars may be sterile, while the remaining biosphere lives kilometers below the surface. We probably couldn't sample from these depths until we start to colonize Mars, but we can get an idea of how habitable the deep subsurface is. To do this, we'd follow up on the magnetic field readings, which indicated a brine reservoir exists below the surface. Another orbiter could be launched to get a more detailed reading of perturbations in the magnetic field of Mars, and surface probes could also corroborate any data with seismic readings, which can indicate variations in density at various depths that would indicate the presence of a subsurface water reservoir. Data from such a probe would also tell us what kind of rocks the reservoir is in, and thus what kind of chemistry it has and how suitable it is for life. Finally, we can get a better idea of how materials are flowing through the mantle with seismic data, and thus how quickly potential metabolites subsurface microbes could utilize are delivered to their habitat. If it turns out that an environment deep underground is conducive to life on Mars, it could give us more incentive to colonize the planet and finally get an answer to this age old question that has occupied the minds of astronomers for centuries. Hey everyone, I thought I'd use this outro to address something that was brought up in the comments of my other videos, and that's that I often pronounce words a weird way. This isn't because I have a speech impediment, nor am I doing a knockoff of a prominent space-based YouTuber who does. Isaac Arthur! The reason my pronunciation seems a bit off is because I'm an American living in Australia, and have picked up some Aussie pronunciation. So I'll say hia versus here, aga versus agar, data versus data, methane versus methane, algae versus algae, H versus H, Z versus Z, etc. Also, if you look at the way I spell some words, I'll often use S where Z is in American English, and that's because the standard American dialect of English uses Z a lot more than others. So that's what's going on. Australian pronunciation with an American accent. As always, if you enjoyed what you saw, please like and subscribe, and share it with friends with similar interests. Thanks.